So, everyone, welcome back to session 3 of Epigenetics, or Epigenetics as we call it. And today we'll be covering a few topics such as that you'll be seeing. I cannot talk a lot about it. Just a quick reminder that please, all the assignments and the quizzes have been emailed to you, and along with all the links of all the previous recordings, feel free to go through them and reach out to us with any doubts. And during the session, we would really like if you guys could maybe unmute or put in the chat box any questions that we ask, and if you want to ask anything, so please feel free to utilize that. And over to Suhana to start the session.
Hi everyone, I'm Suhana and this is session 3, and I hope the previous sessions were full of learning for you guys. Just like last time, we will start with discussing the assignment which was to be completed before the session. So if that can be presented. So if we start with the assignment, the first question said, what is the rule that describes the flow of information from DNA to RNA to proteins? And the answer is option A, central dogma. The second question was, name 3 types of RNA. And according to the options, I think the answer is pretty obvious. It will be option C. Question third asks, how many kinds of bases are there in the DNA? And the answer is 4, which are namely A, T, G, and C. Question 4 asks, generally DNA methylation dash gene expression. And the answer is option A, which is the answer here is option A. Now question 5 asks us, tightly wound chromatin is known as, and the answer is B, heterochromatin. Question 6, the organelle that is responsible for translation is D, ribosomes. Question 7, a sequence of 3 nitrogenous bases that become an amino acid is known as. It was taught in the first session, and the answer is B, codon. It is one of the characteristics of the genetic code as well. Question 8 asks us, histone acetylation and methylation are reversible. And the answer is true, they are reversible. Question 9. Different coat colors in calico cats are examples of dash. The answer to is X inactivation. The calico cat displays a mixture of red and black based colors, depending on which its 2 X chromosomes have been inactivated. Question 10 asks us, X inactivation takes place in, and the answer here is C, either maternal or paternal. Question 11, IGF2 is expressed. The answer is A, in maternal side of the chromosome. Question 12, air pollution can cause epigenetic changes, and the answer is true. Question 13, if C pairs to G in the DNA, what does A pair to? It pairs to T, the option B. Question 14. U is found instead of what base in the RNA? It is B again, T. Question 15. How many amino acids are there? And the answer is B, 20. And lastly, guess the percentage of methylation in our DNA. And the answer is D, 80 to 90%. This were the answer key of the assignment that was provided. And today we will be talking about the application of epigenetics in the field of agriculture, human health, psychology. Now over to Bhuvan.
Hello guys. So as Suhana said, we'll be talking about the applications of epigenetics today, now that we have talked about the basis and the mechanisms. So we'll start with how epigenetics is used in agriculture. So first off, did you guys know that plants also have an epigenome? Were you guys aware of that?
I knew that.
Okay. Okay, so since we're talking about epigenetics, Plants also have an epigenome and undergo epigenetic changes. So, okay. Can you guess other organisms and plants which also have an epigenome? Animals and plants. And other than that, do bacteria have epigenome? Or do viruses have an epigenome? Let's do one thing. I'll show a picture on the screen. What can you guys guess what this is? Okay, so since no one answered, this is the coronavirus. I think you should have known this. Okay, what is this? Can anyone guess what this is? Okay, so this is a bacterium. So plants, bacteria, and virus, in one way or another, do interact with epigenetics. And in the upcoming slide, Priyanshi will explain to you how viruses interact with the epigenome of their hosts and how bacteria have an epigenome. And after that, we can start with understanding how epigenetics is utilized in agriculture. Over to you, Priyanshi.
So here you can see two pictures in two different corners. So can you recognize these pictures? Yes. Any answer? Any guesses? Okay. So we will be talking about epigenetics in COVID-19 and bacteria. So here in viruses, since epigenetics is lesser studied field, so epigenetic changes haven't been found in their genome. But when viruses enter the genome of their host, they make changes to the host genome in order to cause diseases. I hope you know the meaning of host here. Yes. So host are the organisms. Okay. So I think you are aware. And I would like to tell you one more fact that viruses cannot just have DNA as their genetic material, but they also have RNA as their genetic material. And epigenetics in our bodies is important to carry out different life processes such as life cycles of the cell, stably packing our DNA in chromosomes, and other machineries which helps us to fight with the viral infection. So here you can see the comparison between viruses and prokaryotes. So do you know the meaning of prokaryotes? Can anyone tell? You can unmute yourself and then answer. Yeah, Aditya, you can unmute yourself.
Organisms which have not a well-defined nucleus.
Okay, excellent answer. Rightly said, Aditya. So here, pro means primitive and karyon means nucleus, which means the organisms who do not have well-defined nucleus, they are known as prokaryotes. So as we have just learned that prokaryotes do not have well-defined nucleus. So now can you guess where the genetic material of prokaryotes enclosed? Is there any specific place? Can you guess?
Nucleoid.
Okay, well tried. So the answer is since they do not have any well-defined nucleus, so the genetic material of prokaryotic organisms they are not they are not placed in any of the specific place. And now we can see on the slide that viruses they can have either DNA or RNA as genetic material, Whereas prokaryotes, they can only have DNA as their genetic material. And if you want to go further deeper into it, then you can always reach out to us for any further queries. So we will be clarifying it in the next session. Over to Bhuvan.
Okay, thank you, Priyanshi. That was a very nice side information uh, about how other organisms use epigenomics. Now let's start with why we need to study epigenetics in plants, right? So I'll need you guys to unmute yourself and answer. First off, how much is this? This graph shows the growth of population from 1950 to 2100, the year. So how much population, according to the graph, does the world contain in 2020? So what would be the first lower red circle here?
$7.9 billion.
Yes, approximately, yes. And how about the other circle? The one that's placed above the other one. So the other circle shows how much population we'll have in the world in 2050. So how much would that be? The answer is pretty obvious. It's also written in the text.
Around 9.8.
About 9 billion.
9.8
Yes.
billion.
Okay. Great. Great, guys. So yeah, so as we can see, in less than 30 years, our population will increase from approximately 8 billion to 10 billion. That's 2 billion, right? So apart from that, with the advent of technology and better healthcare system, the life expectancy of humans, which means the length of the life of humans, has increased by more than 6 years between 2000 and 2019. In 2000, it was 66.8 years on average. So an average person lived for 66.8 years, whereas in 2019, an average person lived for 73.4 years. So this means that the death rate is decreasing, right? Now, along with that, the birth rate is also increasing, because if the healthcare system is good, obviously children won't pass away upon birth as they used to in ancient times, right? So these two factors tell us that the population is ever increasing, and for an increasing population, we'll need more food. Now, food for more crops, we are going around and clearing, rainforests to convert it into farmlands. It doesn't just impact the world in terms of air pollution or other things, but instead it also greatly impacts the biodiversity, and one could lead to another, and a domino effect could take place, in which we could lose a species that's very important in our ecology, and it would disrupt the ecological balance. So what we need is a solution that helps us make crops that are better in quality, as well as crops that can withstand the changes in temperature and the climate change that's taking place. So let's take a look at why our crops have a problem in them, right? So let's initially assume that when the prehistoric humans started domesticating crops, for example, wheat. Let's take wheat as an example. Now imagine all these candies, all the different colored candies, is the wild population of wheat. So all different types of species of wheat are present. Now, tried and tested different qualities of wheat. And they had to see what they could eat and what they couldn't eat. So they found a type of wheat, let's call it red wheat, because the red ball, and they found a type of wheat that is very tasty, very nutritious, and they really liked it. So what did they do? They cultivated that wheat. And when they cultivated that particular kind of wheat, This means that the population, the genetic balance that was there in the population in terms of number, got disrupted. I'm sorry, guys, am I audible? Could anyone answer?
I think your voice is a bit cracky.
Okay. I'll just quickly connect to the other Wi-Fi. So just give me a
Yeah, that
second.
works.
Hello, am I audible now? Is it better?
Yeah, it's better.
Okay,
Bhuvan, you might repeat a bit.
sorry, what?
Please repeat last 5 minutes, I guess, because it was a bit cracky for all of us.
Okay,
Okay, thank you.
we can do that. So, okay, let's assume that all the colored candies are the wild population of wheat, and all the different species are present everywhere in the forest. Now, a prehistoric human picks uh, one type of wheat that contains a particular DNA, obviously, one breed of wheat that is beneficial to them in terms of in terms of taste or nutritional value or anything of the sort. Now, that human takes this particular kind of wheat and it cultivates it to feed its society. So this now when it's repeatedly cultivating that wheat, it's going through many and many generations, and what's happening is that the genes, it's the human is selecting the good wheat from the bad wheat, right? So over generations, over repeated breeding, what happens is that it concentrates the genes that are useful to us, but some other genes that are also useful to the wheat in order for the wheat to thrive in the environment, they get silenced or they get shut off, right? So because of repeated breeding and trying to concentrate the presence of useful genes that are useful to us, we actually ended up losing the genes that are useful to the wheat in order to uh, strive in the environment. So in the next slide, we'll be talking about the kinds of stresses that there are, because we need our plants to be strong enough to tolerate stress, because now climate change is taking place and the environment is harsher than ever. So we need our plants to be stress tolerant, right? So let's take a look at the different kinds of stresses. And again, I will need you guys to unmute and tell me what this picture is. What kind of stress is being shown in this picture?
Drought.
That's great, Aviral. Yes, this is drought stress. And what's happening in the next picture?
Insect infestation.
Yes, that is also a kind of stress, because when insects eat away plants, obviously the plants won't really like it and can't thrive in an environment like that. And what's happening here? Okay. So the plant is undergoing some kind of disease or some deficiency. So the third picture actually shows a disease which is called blight of potato, and it is caused by a fungus of a kind. So these are the types of stresses. Abiotic means non-living natural phenomena. For example, drought, salinity. Salinity means excess concentration of salt in the soil. And extreme heat and extreme cold are some stresses that our plants need to learn to tolerate with the change in climate. The other kind of stress is biotic stress, which we talked about in the last two pictures. So the first one is pathogens, fungus, bacteria, virus that infect the plant and cause diseases. And the other one is insects and pests, things that eat away our plants. So to overcome these stresses, we need to understand not only genetic but also epigenetic phenomena in order to change or modify the plant's genomes so that they can thrive better. Now, until now, what we have been doing with biotechnology and genetics is inserting a particular type of gene that helps the plant in overcoming these stresses. With epigenetics, what we realize is that plants, we don't have to insert any gene, but we can just turn on the genes that help in tolerating stress. But the problem with epigenetics is that it's not necessarily inheritable. It's reversible, and only some changes get inherited. And that is why we need to further study epigenetics in more depth. Now, let's take a look at how the epigenetic mechanisms in plants take place. So it's a very content heavy slide, but it's okay, I'll walk you through it. First of all, in nature, plants control various natural processes using epigenetic modifications. Processes like what time they'll flower, processes like senescence, and senescence means growing old of the plant, and processes like gametogenesis. So plants control various life processes, even other than these three, to function. And they control these processes with epigenetics. Now, some major epigenetic changes get inherited to the next generation. And this is called transgenerational memory. Trans means across generations. So this epigenetic memory travels across generations. Now, the diagram on the right shows how the events that take place in our plant, which would lead to epigenetic changes being passed on to the next generation. So the column on the left shows what's happening on a genomic level, whereas the column on the right is showing what's happening as a phenotype, or what's happening in the plant. Now, this figure you guys can read on your own, and I will explain this in the next slide. So let's go there. So let's assume that this thunderbolt is a stress that the plant is taking. So the stress could be heat, cold, UV light, drought, salt, nutrition, or any pathogen. So now when a stress signal is given to the plant, here is what was shown in the diagram for the same. One of three things can happen. Either this gene was already methylated. Now I'm talking about methylation because it's the most major and widely studied epigenetic change that we talk about. So I'll say methylation. So either this gene had already been methylated and it gets demethylated at the trigger stress. So either the loss of epigenetic tags could take place. The other thing that could happen is the gene could get transiently methylated, which means that this epigenetic change is reversible. So when the plant recovers from stress, the epigenetic change gets removed. Then the third possibility that could take place at a stress signal is a permanent epigenetic change could take place. And this permanent epigenetic change has the chance to get inherited across generations. Now here we can see the gene has been silenced because of hypermethylation. The red arrow signifies silence. Now let's say it does get inherited, right? Transgenerational inheritance. So when it gets inherited transgenerationally, again, one of two things can happen. Either this gene, this epigenetic tag, is inherited stably, which means it is present in the same form as it was present in the parent. So this gene is off from the beginning till the end of the lifetime of the child plant, right? So the other thing that can happen is an adaptive change, which means if the offspring plant undergoes a trigger environment, which means the same stress signal that in which its parent plant got epigenetically modified, then this gene would stop working because of epigenetic changes. So was this clear to you guys? If you have any questions, you can feel free to ask, and I or anyone else will answer them. Now, we have talked about epigenetics in plants and how we can study them and why we need to study them. Now, let's take a look at epigenetics in human diseases. Now, before we do, we now have Arushi, who will explain to you the basic concepts, or a recapitulation, one can say, about the cellular processes that take place in epigenetics, like methylation and acetylation. Over to Arushi.
Thank you, Gaurav. Hi guys, time for some revision. To further learn more about how epigenetics plays a role in disease progression, let's first recapitulate the cellular processes, as Bhuvan said, that take place in the epigenome. However, before we do that, let me ask you, what do you guys remember about the DNA methylation? Anyone want to summarize? All right, that's okay. So, well, first I'll start with DNA methylation. It is one of the most common and widely studied epigenetic change, or say modification. This takes place mostly in the cytosines that are next to the guanines, C's and G's respectively in the genetic code. It consists of addition of a methyl group, but how does that happen? The enzyme DNMT, which is the DNA methyl transferase, as you can see, the term transfer is present in the name itself, helps in the transfer of methyl groups to the DNA. DNMT is a group of enzymes, so different DNMTs do different kind of transfers. The part of DNA that is not methylated is active, and hence transcription or gene expression takes place in the non-methylated regions of the DNA or our genes. Transcription is a process of copying a segment of DNA into RNA. The RNA in turn helps in making proteins, which leads to gene expression. The figure here shows replication. We can see one parent strand in each daughter DNA, and that it is half methylated. And as you can see, the DNMT one helps to complete the entire process of methylation. Now, as we've studied, histones undergo two types of changes. First is methylation, which has been explained, and second is acetylation or deacetylation, which is a new term and will be discussed in a while. But does anyone remember what acetylation is, as it has been discussed in the previous session? Anyone? Yes, you're absolutely right. So now let's begin with the process. In the bottom right figure, we can see a few chemicals. These chemicals are added to the histone tails. When methylation occurs, it forms heterochromatin. When methylation occurs, it forms heterochromatin, and it tightens the DNA, as we can see in the top figure, which leads to gene inactivation, and due to this, genes cannot be expressed. During acetylation, HAT, which are histone acetyl transferase, just like we talked about the DNMT, it helps in acetylation by forming euchromatin and opening up the DNA, as you mentioned in the chat, and because of which genes are able to because of which genes are available for transcription and hence expression. Deacetylation, which is a new term here, is the process of removal of the acetyl from the tails, which is done by HDAC, which is histone deacetylase. The term itself literally means that it deacetylates histone, hence removing the acetyl from the histone tail. Now I have a question for you all. If acetylations form euchromatin and hence loosens up the DNA, what do you think deacetylation does? Anyone can go, you can guess, it's all right.
Will you repeat?
Yeah, sure. So if acetylation forms euchromatin and loosens up the DNA, what does deacetylation do? What do you think it does?
It tightens it.
Yes, you're absolutely right. Even Rakshit, you're right. It tightens it back up. So basically, the opposite of loosening up, that is tightening up. So that was it for my part. Now I will give it over to Bhuvan. Thank you.
Okay, I hope your basics have been cleared out, because now we'll be talking about diseases. And again, I need you guys to tell me, why do you think we need to study epigenetics in diseases? What could we do with the knowledge of epigenetics in diseases?
To find out why a certain disease affects our body in a certain way.
And then what we can do once we have found out why that disease has impacted our body?
To look for different kinds of treatments. So,
Yes, we can make our treatments more effective, and that is the main reason why we need to study epigenetics in human diseases. So now the text here says that epigenetics is a major part of almost all the processes that take place in our body. For example, histone methylation, as we studied in the previous slide, histone methylation mediates major processes. For example, repair of DNA, cell cycle, which is cell division, division of cells into more cells, responses to stress in humans as well, helping us in development, cellular differentiation, also aging. So, histone methylation is a part of all these very important processes in the cell. And any change in any epigenetic signal or regulation can be pathological, and it could lead to any kind of disease. In the upcoming slides, I'll be talking about neurological diseases, cardiovascular diseases, metabolic disorders, and Kartavya will talk about cancer development. Therefore, identifying drugs that inhibit the epigenetic changes gone wrong are also of great clinical interest. So, yeah. So, first we'll talk about neurodegenerative disorders, which means diseases of the brain. So, I'm pretty sure you guys have heard of Parkinson's diseases. Does anyone know what happens in Parkinson's diseases?
Half of the body stops functioning.
You're kind of correct. Let's take a look at one basic observation first. So, as we grow, as we age, our genes undergo gradual and continuous methylation. So, with age, our genes kind of start getting gradually methylated, right? So, let's remember this. Now, what happens in Parkinson's disease is that in the area of our brain called midbrain, some neurons are present which synthesize dopamine, which make dopamine. And dopamine is a hormone which helps us feel happy. So, technically, so dopamine synthesizing neurons in our midbrain degenerate. So, we suffer a loss of dopamine synthesizing neurons, and this is the main feature of Parkinson's disease. Other than that, another thing is seen which is called dementia. In dementia, what happens is that a kind of a protein called alpha synuclein, and from now on, I'll call it alpha syn. Uh, Alpha syn gets accumulated in our neurons in the form of these darkly stained bodies called Lewy bodies. Now, this is not a cell, this is just the accumulation of protein amongst our neurons. All these tiny dots in the network of light pink lines that you can see are neurons. And Lewy bodies, which are comprised of alpha syn, have been deposited here. So these are the two main things that happen in Parkinson's disease. Another thing that was recently found out related to epigenetics, of course, is that DNMT1 goes from the nucleus to the cytoplasm. Now, remember the fact that when we age, we need more methylation in our DNA. But if we are old and we have Parkinson's, what will happen is that DNMT1, which helps in the methylation of our DNA, goes away from the DNA, away from the nucleus to the cytoplasm of the cell. This would lead to a major hypomethylation event taking place in our nucleus, which is obviously abnormal, and that is one of the reasons that Parkinson's disease takes place. So, hence, there is a global hypomethylation in the neuronal DNA. Global means throughout the DNA, hypomethylation means low levels of methylation. So, I'm hoping all this is clear to you guys. So, the three things that go wrong in Parkinson's disease is that 1, we lose dopamine synthesizing neurons. 2, alpha syn gets accumulated in our neurons, around our neurons. And 3, there is a global hypomethylation in the DNA. So, these are the things that go wrong. And if we can maybe find a cure for it, we can use epigenetics to administer it, right? So, the next type of disease that we will talk about are cardiovascular diseases. What happens in there's one particular disease that I have included here. It's called atherosclerosis. What happens in atherosclerosis is that our arteries, these are our arteries, gets narrowed, and they get narrowed because inside their walls a type of substance gets deposited, which is called plaque. And plaque, I'm sorry about that, and plaque is a mixture of calcium, cholesterol, and fats, and this builds up inside the artery walls. It narrows the arteries and thereby constricting the flow of blood from one place to another. And sometimes our arteries get so narrowed down that the blood can't pass through this at all, and the blood starts clotting in our arteries where it's supposed to flow, and that's obviously a very scary phenomena that takes place. So this happens because of the hypermethylation of a gene called ESR. It's called estrogen receptor, and this is an atheroprotective gene, which means that ESR helps in protecting us against atherosclerosis. Now, this estrogen receptor gene, because it says estrogen, is present in women. And do you guys remember, or have you guys heard that older men are more susceptible to heart diseases? Have you guys heard of this myth? Okay, so that's a myth, as I just stated. Women are also pretty susceptible to heart diseases because as they grow old, their ESR gene gets methylated. Because we just read about the fact that our genes get continuously and gradually methylated as we grow old. With the methylation of ESR gene, this means that the ESR gene expression is lowering. And when the gene expression lowers, there is lesser and lesser protection against atherosclerosis. And that makes us more susceptible to atherosclerosis. So with age also, people get atherosclerosis. And other than that, if someone does have atherosclerosis, it is seen that their ESR gene is getting hypomethylated. Yeah, so then metabolic disorders. So metabolic disorders generally are results of environmental changes which impact our gene expression due to epigenetics. So in the previous classes, do you guys remember talking about how environmental changes or changes in food have impacted some organism to change its coat color and become obese and have diabetes? If yes, what was that all about? What was the name of that case? Anyone? Okay. So we were talking about agouti mice. Remember how two mice that were identical twins were so different from each other because of how much nutrition they were given in the earlier stages of their lives. So metabolic disorders like diabetes, hypertension, obesity, and PCOS can have other factors as well, but environment is also a major factor that plays role in the progression of that disease. So these are the three major kinds of diseases that are impacted by epigenetics and that can potentially be cured using epigenetics if we study epigenetics in more depth. Over to Kartavya, who will now talk about cancer.
Thank you, Bhavan. Before starting the session further, uh, if you can if you can remember, in the previous session we had discussed about genomic imprinting. If you can recall, in the previous session we have introduced the topic genomic imprinting. Can anyone tell me what genome imprinting is?
Process in which one copy of a gene gets silenced.
Uh, can you repeat?
A process where a copy of a gene gets silenced.
Yeah. And about the expression, what type of expression do you observe in humans? Is it biallelic or monoallelic?
Biallelic.
Great. So, as been as Avel just said, genomic imprinting is a epigenetic phenomena where gene expression is regulated in parent of origin specific manner. That means if you can see the diagram, the gene A from the parent gets the promoter of gene A from the maternal chromosome gets methylated. Thus, we don't see an expression of gene A, but gene B still has an expression in maternal chromosome. Similarly, in the case of parent, gene A is well expressed, but gene B gets imprinted. So, anybody can tell me what type of phenotype would you be observing in this? Would you be observing both A and B genes or just a single product? Any guesses? Uh, if no, both both alleles A from the maternal chromosome and B from paternal chromosome both will be expressed, and we would be getting both A and B proteins. Thus, this is a normal situation. It happens normally across all the individuals. But next, let's take another case where suppose there is a mutation in maternal chromosome or there is a deletion, there is some mutation in maternal chromosome. Thus, we don't see usual expression of protein B. So, what would be the phenotype in this case? What would be what would we be observing here? Can anybody guess? Not paternal. I was asking what specific protein. Yeah, great, Shefali. We would be observing only expression of gene A, and we will be getting protein A. This situation is results in certain disease. Similarly, in the next, if we delete, if there is a deletion paternal chromosome, what would be the end product we would be observing? Uh, I guess no answers. We would be observing B only, and that's how any mutation in or deletion in any of the uh, genome imprinted gene get causes any disease to occur. Particularly, there have been identified several genes which are imprinted, as we have discussed in the previous session, where we have discussed about IGF2 gene. Today, we shall be discussing about two important diseases: Prader-Willi syndrome and Angelman syndrome. Both of these diseases occur due to genomic imprinting in in chromosome 15 locus 15q11. That region, that genomic region encodes the protein essential for both of these genes, and there are total three proteins which are found to be acting during these situations. And if any mutation occurs in either of the parent syndrome, if suppose there is a paternal deletion, we would be observing Prader-Willi syndrome, and if there is a maternal deletion, we would be observing Angelman syndrome. The major the major takeaway from this is that both of the genes are encoded the same genomic area. One causes you to become obese, and other causes you to become thin, and both have mental retardation. Let's move further. Now we would be starting about a new thing, which is cancer epigenetics. So can anyone tell what a cancer is? You can unmute for answers.
Abnormal cell growth in high amount.
Yeah, there is uncontrolled growth of cells. The enormous cell division resulting in abnormal growth, and then we would be observing the cancer phenotype, that is an increased number of cells. A cancer has approximately 7 hallmark features, which basically uh, informs the observer that it is a cancer growth. First is tissue invasion and metastasis, inflammatory microenvironment, insensitivity to growth inhibitors, cell sufficiency to growth signals, sustained angiogenesis, evasion of apoptosis, and limitless replicative potential. All these factors uh, are responsible for making a cancer cell and its progression. The important here is that that when the normal cell divide, they have ability to understand their environment. Like if if one cell divides and another cell is also dividing, and both reaches approximate distance to each other, there is cellular signaling which coordinates that we should stop division, and thus the cellular normal cell don't divide abnormally. But in the case of cancer cells, this property is diminished, and that's why uh, even after contact inhibition, even after the contacting, the cancer cell doesn't stop growing. Mechanism of gene silencing in cancer. Like we have already discussed so many times here that methylation in methylation causes gene to change expression patterns. There is a differential expression. So we can say that cancer progression is controlled by tumor suppressor genes. The tumor suppressor genes are those genes which regulate cancer formation. If they are active, that means you won't be seeing cancer formation. But if due to certain factors like any addictiveness or any new any type of nutrition you take or any other environmental factor, the expression of gene changes as it gets methylated and thus tumor suppressor gene gets repressed and we see tumor progression. Other than methylation, histone modification in cancer cells also plays an important role. Can anybody tell me uh, uh, what would be the what would you be observing if a cancer cell has a lot of histone modification? Um, I don't think there's an answer. So, in case of a normal gene, which is it has a enhancer region which regulates, there's a promoter region, and there's a gene body which has information to which protein is being encoded. In that case, if there are histone modification, then the gene gets repressed and chromatin modification changes resulting from euchromatin to heterochromatin. Thus, we get a silence gene, and thus that's the reason why tumor suppressor genes doesn't work in cancer cells. Similarly, in case can you think of any repetitive sequence if you know about any? I guess no. Uh, our genome is filled with a lot of genes, a lot of genomic information. Some are stringent, some genes occur only once in our entire genome, which are required for specific purposes. But some genes which are necessary for various purposes like the tumor regulation, controlling tumor cells, tumor progression, and certain other important functions like translation of protein or DNA methylation and any other function. In that cases, we have a repetitive sequences which are present, and these repetitive sequences leads are turned off in case of any these are initially turned off in normal cells, but when the change in histone occurs, these genes these genes can get activated, and hence there is a lot of change in other neighboring genes, and that is accompanied by loss of repressive marks. Like as we discussed earlier, there is acetylation gets decreased on histone, and active active it gets activated. The gene gets activated and chromatin opens, and we can see expression of incorrect genes. Incorrect genes means the genes which initially shouldn't have been expressed. Like there are certain types of oncogenes which causes cancer development. If those genes get activated, there's sure 100% chances that you can get cancer. Since in the previous slides, we discussed about various hallmarks of cancers. if you can see that each of these hallmarks is are regulated by different genes, and changes in changes in expression of any of these genes can change the phenotype of a person. It can change a cell from normal to cancer, and especially the P53 gene or the genes involved in replication, like cyclin P16. These are very important because that regulates how many times a cell will divide and whether a cell would undergo death or not. If cell doesn't go under death or or repetitively divides itself, then there's chances that you might get a cancer. So, can you tell me what is being shown in this image? Can you tell what is shown in this image? I think no answer. No, no, yeah, it's uh, it's not DNA replication in full, but we can say that it's DNA methylation. Once, let's imagine that when our DNA gets replicated, we have certain types of epigenetic marks. But how does it get passed on to the cell, to the DNA which has been newly formed after replication? That is done by DNMT, that is DNA methyltransferase. And currently, if DNMT activity is not stopped, it can lead to certain types of diseases, genetic diseases which we haven't seen before, or which are expressed very low, or which are observed very low, like Parkinson's disease or any type of diseases like Prader-Willi syndrome or any other diseases which you might haven't heard of even. For this, a, com- a company thought to block DNMT activity, and they gave and they produced a cytosine analog which is 5 as a cytidine. This drug acts as cytidine analog and exchange exchanges the cytosine in the DNA in the DNA which is being targeted, and hence since DNMT doesn't recognize any chemical other than the distinct cytosine, it won't add a methyl group to it. And this drug has been recently approved by FDA and is in the market. Can you tell what is FDA and what is its role? Can you tell what is FDA? Yeah, well, you're right. FDA is the Food and Drug Administration agency based in US, and it's the oldest and the most proactive drug regulating agency. If any drug gets passed by the norms of FDA, then it means it has reached that result, or it has reached that final stage where it can be uh, delivered to other nations as well, because most of the countries, even other having regulatory bodies other than FDA, like in India, we too have National Drug Agency, which regulates the formation of vaccine regulation, and that also works behind the FDA. Once FDA has given uh, its property to the drug, then it means that other agencies can think that this drug is fine and will work fine. Let's see a video here which is based on epigenetic therapy. Is it audible?
Not audible.
I'm sorry for the trouble.
It's not audible at all. Now it is audible.
This video is brought to you by the Epi Femcare project team and focuses on epigenetics and cancer.
It was audible now.
Yeah. Sorry for the trouble.
This video is brought to you by the Epi Femcare project team and focuses on epigenetics and cancer. It's been known for some time that some cancers can occur because of genetic mutations, changes to the sequence of DNA within our genes. In breast cancer, for example, mutations in the gene for p53 result in the loss of cells' ability to repair DNA and can contribute to cancer development. But it's becoming clear that it's not just genetic mutations that contribute to cancer development. It's also how our genes are controlled. Epigenetics literally means on top of or in addition to our genes. The Epi Femcare project is focusing on the most common epigenetic change, DNA methylation. This is the addition of a CH3 molecule or methyl group to the DNA. Epigenetic changes can control whether our genes are switched on or off. Erroneous epigenetic events, such as too much methylation at genes important for maintaining healthy cells, are very much a feature of cancer. As a tumor grows, DNA is released into the blood and can be collected easily. The Epi Femcare project will use new technologies to identify differences in patterns of DNA methylation between women with and without cancer. We will develop a non-invasive cancer detection test that finds these cancer-unique epigenetic changes. Our aim is to improve on current methods of screening, diagnosis and personalized care, including the CA125 test used to help diagnose and monitor ovarian cancer. The Epi Femcare test will be both specific to breast and ovarian cancers and reliable, thereby improving clinical care for women with these cancers. The Epi Femcare project is partially funded by the European Commission's Framework Programme 7. The next video explores the project in more detail.
Let's now talk about psychology and epigenetics. As you have heard from our previous sessions that psychology and epigenetics are interrelated. Whether it is your parents' behavior towards you, your learning and memory, your childhood experiences, or the nutrition you take, all causes certain epigenetic changes, resulting in change in gene expression, and that defines how you progress in your life and how you will be in the future. About talking about early childhood experiences, can anyone tell me what type of childhood experiences uh, a person can observe for a healthy epigenetic? Anybody can try? Yeah. Yeah, thank you, Avril. Other than good nutrition, a close degree of positive attachment from the parents, or, or a good social life, a good nourishment during the prenatal stage also defines how you will be in the future. Early childhood experiences are important because if your childhood is pretty good, I mean that your parents treat you very well, you have a good, healthy mindset, then it would definitely strengthen your biological defense system. That is, it will build up your immunity. It will give you a reproductive success, and it will also be helpful in your future as it will give you adequate parenting. Now, there's an old saying, you are what you eat. Any nutrients which you eat, as we have discussed in the previous session where we discussed about nutrition during famines, we have discussed that nutrients can also modify epigenome of a person. It can change the expression of critical genes associated with physiology and pathology, and it can also change the expression of genes during embryonic development. It can either decrease your aging, meaning it will make you be healthy, or it can also lead to cancer. There's certain, it has been observed that DNA methylation and histone modification can alter uh, can alter enzyme enzyme activity, and availability of substrates for enzymatic actions can also change your epigenetics. In in an experiment, uh, there was an experiment which showed that rats fed with a low diet of methyl groups or low methyl diet showed reduced expression of DNA methyl transferase, and it also showed that they had decreased expression of glucocorticoid receptor, which is essential for glucose metabolism, and thus those rats were found to be obese, and a few of them were observed to be mentally retarded too. Can epi- uh, like can you tell me, can epigenetics be influenced by addiction too? answer would be yes, because any type of addiction, whether it is for drugs, alcohol, or for any other activity like betting or any sort of other addiction, can change your epigenetics. If your It has been observed from various experiments that a father or a grandfather who had been addicted to drugs or alcohol had showed that their children or their grandchildren too were seem to be addicted to that behavior, and those types of and that leads to various diseases like autism, schizophrenia, ADHD, and addictive behavior. When we talk about addiction, the one thing we must talk about is smoking, because tobacco is used to health and tobacco is highly addictive. Certain experiments carried on bronchial cell culture where they treated bronchial cells with liquidified smoke. A fascinating thing that smoke can also be turned into liquid. Can you tell how how can it be possible that you can turn smoke into liquid?
Now, I have a question, Kartavya. Can you hear me? Vinita
Yes,
ma'am.
yes, ma'am.
Like just now, I was hearing your session, and I heard that there are some diseases or some behavior which is hereditary because of it. So you said that there are like schizophrenia and then four, five more diseases. Does it mean that these are hereditary? All these diseases which you mentioned. Over to you.
Yes, ma'am. These various studies have shown that these diseases can be hereditary, but it also depends on the several factors. It also depends on how how the child is behaving to the environment near to him or her. If from the childhood you are staying away from these things, then it would be very less. There would be very less chances that you will adapt to that behavior. But in the
No, but
same
my
case,
question, I'm sorry, but my question is nowadays because of stress, many people are getting this. So, so is it always hereditary or it is also related with the environment, stress, etc.?
uh, it's related to environment too, ma'am. The genes do control these behaviors. Like there are certain genes which control your cellular activity in the mind. Like Bhavan mentioned about neurodegenerative diseases. Genes control these certain activities in synaptic in nerve cells. If you or if there is gene impaired in these cells like nervous cells, then it would be possible that you can develop these diseases. And that's why most of the children, if born with, uh, if already born with these diseases, are are we can say that that is genetic. But in similar thing, uh, over the if your childhood experiences are bad, like I mentioned, it, it depends on your childhood behavior too. If you have been treated well, if there has been if you have been treated positive, then these changes may be very minimal. The genes may not be altered at all. But if the
Okay,
genes
thank
have already been changed during your uh, gestation period or during child development, then we can't change them.
okay, thank you. Thanks a lot.
So we were talking about smoking, And can you tell me how one can change smoke uh, from gaseous state to a liquid state? I think there's no answer. Like you have, yeah, compressing it is the best answer. Pressure and temperature, yeah, yeah. All these answers correct. The best evaporation and condensation are the best ways to convert a gases into a liquid state, and that's how they done they did for this test where they found that there was change in expression of EZH2 gene and methyltransferase gene, which then change the expression of certain oncogenes like BMP3, SFRP2, and hence there was a change cause oncogene signaling, which may lead to lung cancer. Like ma'am, you have asked the question. Epigenetics is related to your behavior. If if in an early stage you are treated with goodness, then your chromatin would be open. The genes would be expressing itself regularly, and you will be having a healthy mind, far from mental disability. But in case of depression, it has been found that the, the histones and the chromatin structure develops into uh, chromatin structure gets altered and forms spiral loops, and that has been observed in the case of depressed patients. There is change in neurotransmitter pathways and certain genes in responsible for brain development, and thus we see depression.
Uh, is there some background noise? Audience, please mute yourself when Kartavya is speaking.
Yeah. So, any questions please here? If no, we would be jumping into the so, next slide. Before that, I have a few questions for you. Uh, if you can uh, scan the QR code.
Alternatively, the link is also put in the chat box if you want.
Excuse me. I have a question regarding the cancer treatment.
Oh, yes, you can go ahead and ask.
Yeah, so for example, they said that they can treat the cancer cells by changing the epigenetic by testing your genes. So at that point of time, they will only be able to test the genes which they have taken the sample for. How will they change each and every gene in your cell or change
Actually,
the demit?
uh,
Yeah.
actually, it was just a testing kit. They were planning for testing kit for uh, identifying the change in methylation, so that they can know whether what are the changes in the gene. They have there are markers for almost every gene. There is a database specially created for cancer genes where they have put all the information on methylation and other things.
Okay. Thank
I hope most uh, Absolutely, memory and learning can be affected by epigenetics. We have one no problem with uh, epigenetics and can affect your memory and learning too. We would be learning about shortly. If all of you have answered this question, let's go to the next question. You can raise your hand if you have done it.
Kattavya, please keep track of time, bacha.
Yes, ma'am. Just two more slides are left from my part.
Yes.
Uh, I hope all have answered. Let's go to the next question. Which part of your brain would be responsible for keeping track of memory and learning? For your easiness, I have added a diagram of brain. You can think and respond. Yeah, if all the responses are made. Yes, hippocampus is the region present in gene which is responsible for keeping track of your learnings and memory. It controls all of your thoughts, learning, and memory you have garnered from years. And that's why if anything happens to your if during any head injury, if there's an effect on your hippocampus region, then there's slight chances that you may face short-term memory loss or long-term depending upon the grimness of the head injury. Okay, let's let's move to let's move to a final thing, and we will be discussing about epigenetic regulations of learning and memory. As we have talked that epigenetics to regulate learning and memory. It has been observed that changes in histone in- influence long-term memory accessibility and expression of genes relevant to learning and memory. There have been identified several genes which have been methylated, showing that showing that it can affect cognitive function and mental health. Depend, Epigenetic mechanisms can influence genomic activity in brain to produce long-term changes in synaptic signaling and organization, even morphology and supporting learning and memory. Based on the few examples, there are two major examples which I have come across. One is Rett syndrome and other is Rubinstein-Tybee syndrome. Don't worry, don't have to go to learn much details about these. We would be talking in brief about Rett syndrome. If you can see the little girl, even from her appearance, you can tell that she might be suffering from some mental disorder. While I should not do that. Rett syndrome is caused due to mutation in gene MECP2. The major role of MECP2 is to read DNA sequence and check for methylation, and then it binds to methylated BT. This causes this causes gene expression changes, supporting dendritic and synaptic development and hippocampus dependent memory. Altered expression of MECP2 has uh, has given the researchers that histone acetylation. There was histone acetylation, neuron cell death, increased anxiety, and cognitive difficulty, including social withdrawal. Such type of patients do uh, do suffer from seizure, loss of motor skills, and delayed speech and loss of communication. Can you ever guess why all the mental disorders causes loss of motor skills and delayed speech and loss of communication? Have you ever pondered about that? Why that only that every disease related to mental health causes change in motor skills and delayed speech or change in speech and communication? If nobody has any answer, then I would say that since entire brain is formed of neurons, then and neurons control the movement, the motor movement of our entire body, then that's responsible for such things. And since hippocampus has been affected, then it it would definitely cause something to your speech and communication skills too. That's all. That's all from my side. I would hand over to Janvi.
Okay, guys. Hi again. So I'm putting a link on the chat box, which is for the assignment for this session. This is your second assignment. All material and all of the material that you'll require to answer this particular question. While the question might look difficult, it's actually very easy. All material is on our in the past two sessions that we have done, and all of these will be uploaded on the KaryoUS website and on YouTube, so you can go through them in case you need any help. And I'd also like to end the session for today. And thank you so much for tuning in for this session organized by Genesis, the Life Sciences Society, and Team KaryoUS. Again, I'd like to thank our presenters from Genesis and KaryoUS for all the hard work that they've put in and to bring out this edition for epigenetics. And as you all might have realized, epigenetics is truly everywhere. It's in humans, it's in plants, it's also in viruses, and it's responsible for a lot of the diseases that we might see around or we might have heard about. And it definitely studying this better will help us adapt our medicines better and get more streamlined treatment. So it is a very important and emerging field. In any case, do write to us in case you have any feedback or doubts, and we'll be happy to help clear them out over there. And we'll see you guys next week for our last session on the 14th of August. Enjoy the rest of your week. And again, do finish your assignments as they will be required to get a certificate at the end of the course. Thank you, guys.
Janvi, once everyone leave, can we just stay back for 1 minute? All the peer tutors, just for 1 minute.